Welcome everyone to this joint webinar organized together by Rocket and Carbohyde. We decided to pick a few topics that are not that often discussed within the cyclodextrin research, but are eventually shaping the future, and therefore we think they are very important to discuss. So we picked a bouquet of biopharma applications and selected a few speakers who, in our opinion, can provide very valuable insight in these topics. So very briefly, the program of today, we'll start with Alain Bluet from Rocket, giving some market insights about this industry and these applications. Then we'll continue with Milo Malanga from Carbohyde, talking about drug delivery systems for gene therapy using cyclodextrins. Then 2 lectures will be held by Rocket. First, Tao Peng, and then Hailong Zheng will tell us more about Vepto's Biopharma and their applications in protein formulations, first in biologic downstream process and formulation development, and then a comparative analysis comparing HPBCD's performance to polysorbates. And then in the end, we'll have a panel discussion with all the speakers and 2 further experts, Carmen Popescu and Katrina O'Driscoll, invited, where we'll have an open discussion about these topics, how we see the presence and the future of cyclodextrins in these areas. You are encouraged to raise questions during the presentations. These you can type in the chat box, or you can, there is a Q&A button also in the bottom where you can ask your questions. If time allows, I'll raise the questions right after the presentation to the speaker, and if not, we'll discuss them in the panel session. So they'll be addressed. And with that, I suggest that we start, and I'm asking LM to share picture, unmute, and kick off the talk.
Yeah, thank you so much, Tamas. And then on behalf of the Rocket team, I would like to stress that we are really happy to make this webinar with Carbohyde team. So I'm going to share my screen, and let me know if you can see it.
Yes, we can see it.
Okay, thank you. So I will put it on mode style. Okay, so here is just a kind of reminder about some definition of what biological drugs are. It's not it's something I'm pretty sure that all the audience know about. Some the type of different biological drugs, and indeed will end up with a market inside in the biological drug sector. So first, I took, what I found on the web. What is biological? And really good to know that it was discovered how many years back by Shibasaburo and Kitasato and Emil van Beering, that, all using the serum from animal to be injected to produce a desired antibody. Most interesting, it can be traced to ancient China, Africa, Turkey, maybe 1000 before Jesus Christ, when smallpox inoculation was used to provide immunity to that disease. And finally, okay, the result, have you heard about? And the most important is they are derived from living cells and indeed developed for blood, protein, virus, and living organisms. Then I took one, I think it's interesting what Wikipedia is saying about biological drugs. First, they talk biopharmaceutical, and they are extracted from living source or semi-synthesized. Most until they give the list that we will see further on. And they mentioned they are composed of sugars, protein, nucleic acid, and a very complex combination of these substances. And most important, again, saying maybe living cells or tissue. And finally, they mentioned at the end that is isolated from living source, either human, animal, plant, fungal, or microbial source, and used in both human and animal medicine. So let's go on the official, let's say, from regulatory agents. So this one is from the EMEA. And EMEA, indeed, you start by saying these are medicine to treat patients with chronic or and in very often disabling condition like diabetes, autoimmune disease, and cancer. And I have a problem. Oh yeah, okay. And these biological medicine contain active substance, again, from a biological source, like always we see the word living cells or organisms from different origin, from human, animal, or bacteria. And they put the word produced by cutting edge technology. So they are expecting that all this technology will continue to improve and to be the state of art of this technology. And indeed, they say they are made of protein and differ in size and complexity in structure. And they gave example like insulin being one of the first, the growth hormone, or a monoclonal antibody. So let's take what the US FDA is saying. Again, you start seeing these medicine to diagnose, prevent, treat, and cure. They are made from large and complex molecule and from a living cell, but they went further on. They said that they are often, very often, which is the case, more difficult to characterize than small molecule or the conventional molecule. And they even went further. Why it is so difficult? Due to the inherent variation, they result from manufacturing process that present challenge in characterizing manufacturing these products that do not exist in the development of small molecule drugs. So they put already here a side-by-side, a quick comparison with small molecule drugs. And I took also this ATMP, the Advanced Therapy Medicinal Product, defined by EMEA. And indeed, you already know the ATMP include the gene therapy medicine by, again, for therapeutic, prophylactic, or diagnostic effect, and inserting recombinant gene into the body to treat a variety of disease. There is the cell therapy, or what we say the somatic cell, as opposed to reproductive cells. Again, these have been manipulated to change their biological characteristic and not intended to be used for the same essential function in the body. And finally, this tissue engineering medicine, again, modified to repair, regenerate, or replace human tissue. And they went even further, and they have the GMP part 4 in the volume 4 of the GMP in Europe is only dedicated to ATMP, good manufacturing practice. Finally, it's very important to talk about biosimilar, because biosimilar is one of the most biggest markets in the biopharma market. So let's see side by side how EMEA defined it and how US FDA. They are, aligned. It is highly similar. It's not similar, but highly similar to another approved biological drug. For both, we call it, in EMEA, they call it the reference medicine, US FDA, the reference product. Taking EMEA, what means highly similar? It's highly similar in structure, biological activity, efficacy, safety, and immunogenicity profile. And it is approved by EMEA, which means has been prior to approved, evaluated according to the same standard for a drug product in terms of pharmaceutical quality, safety, efficacy. So this, because due to the complexity of the manufacturing of the biological, they even stress the word, it is not a generic. A biosimilar is not generic. Generic is only dedicated to small molecule. And US FDA, the highly similar mean no clinical meaningful difference with the reference product in terms, again, safety, purity, potency, and it's mean the manufacturer of biosimilar, he will need to submit all the data to show the quality, efficacy, safety, and the immunogenicity profile. So here it's a comparison, let's say, side by side, reflecting already what the US FDA has stressed. When you take small molecule, example, the most known, aspirin, structure is known, while the large molecule is a very complex mixture. And they are defined by their manufacturing process, while the small molecule can easily be chemically defined and characterized. And small molecules usually are stable to heat. We all know biological not stable to heat. They are even susceptible to microbial contamination, which is not the case of the small molecule. And the biological, so far, they are all produced in aseptic condition because they are indeed sensitive to heat. So, and the small molecule will depend on the route of administration. Small molecule, mostly they are not immunogenic, while the biological mostly are immunogenic. And for both, developing a new small molecule or developing a biological will take at least 10 years from the early discovery till the product launch. So type of biological drugs, it's also, I think, known from all of you. Well, let's talk first about vaccine. Protecting against harm or disease, and vaccine became, let's say, a kind of hot topic during the last 3 years with the COVID-19. Blood, the whole blood or blood component, and the most known are factors 8 and 9 obtained by blood fractionation for hemophilia. Monoclonal antibody, again, it's well known. The recombinant antibody, which are monoclonal antibodies generated in vitro using synthetic genes. Allergenic are part when it comes to the monoclonal antibody, which are directed against some pathogenic such IgE or pro-inflammatory cytokine. The hormone therapy, well known, started with the insulin years back. Then we have the gene therapy that we mentioned just before, the tissue therapy and the cell therapy, and finally the recombinant therapeutic protein using the ADN technologies. So market insight, global biological market is really a growing one. We are talking about 335 billion at the end of 2021, and it's going to grow by 8.5% to reach this 8,017 billion. The key driver, you all know, the rising in prevalence of some chronic disease like cancer, autoimmune disease, and diabetes. These three diseases contribute for more than 60% of the overall biological market. There is, for sure, the development in gene and cell therapy. There is a lot of money on the table, a lot of money in fundings. And just last year, there was 11 billion fundings just for gene and cell therapies. And there is a demand of biologic across the globe. And indeed, it's, as mentioned by the EMEA, cutting edge technology. This means a continuous investment to obtain higher efficiency, higher yield. And if we take these 4 sub-markets that I select, both of them, the 4 of them are growing by 2 digits. We have 10% for vaccine and monoclonal antibody, going to 80.6% for the gene therapy. These are really growing, and the driver, you all know about the CRISPR technology, CAR T cell technology, and there is a lot of funding, as mentioned just before, this 10 billion funding just last year. The vaccine, indeed, there is a rising incidence of re-emergent infectious disease, and it has been boosted, and I'm not saying something new, boosted by the mRNA technology for the COVID-19 vaccine. And monoclonal antibody, indeed, there is a growing prevalence in cancer and other chronic disease like immune and diabetes. And the large market, North America, excuse me, and Europe, but Middle East and Asia are fast growing market. And one word about the biosimilar, because it is one of the biggest segment, and as you can see, the growth is 2-digit, it's almost 18% from 2022 to 2028. Okay, Europe for the moment is the largest and most mature market. We have over 74 approved biosimilar. US is coming. Now they have over 40 biosimilars, but as I mentioned, the other emerging country like India and Asia are also having a biosimilar approval. What is the market driver? Indeed, loss of patent exclusivity, and the other one is the cost effectiveness. It's a huge demand in cost effectiveness and rising, indeed, incidence of some chronic disease. And I will finish with this one, that to show you that in 2018, we had 68% of drug approval, which are small molecule, versus 32%. And last year, there was 52% of drug approval, which are biological. And this ratio will continue to increase in the coming years. And we know, for instance, according to IQVIA, that there will be 10 marketed biological drugs that will be facing competition from biosimilar that are expected to be launched in 2023. So all to say, this biologic market is huge, dynamic, and fast growing. And thank you for your attention.
Thank you very much, Ham, for the nice summary on the market insights and all the biological products. And I must apologize because I haven't done a proper introduction. So Ham is working at Rocket as the regional head of Biopharma technologies, and just gave a very nice presentation. So I'm not missing to introduce the next speaker, who is Milo Malanga, the Chief Scientific Officer of Carbohyde. And Milo will talk about the potential of cyclodextrins in gene delivery. Milo, are you ready?
Yeah, can you hear me, Tommy? Good morning.
Yeah, I can hear you, see you. So go ahead.
Hello, everybody from Rimini. As you can see, I'm enjoying very much my vacation on the beach. And I'm really happy to meet this webinar and be part of it. And thanks, Tommy, for the kind introduction. I will share the screen. Yeah. Mm-hmm. Can you see the presentation, the screen? I hope it's okay.
We do. You can go ahead.
Okay, so let's get started. And this is the outline of the presentation, and I will give first a general concept about gene delivery system, and then go in detail about some specific and selected example from the literature and a kind of state of the art about the potential of cyclodextrin for gene delivery system. So essentially, gene delivery system can be divided in two huge categories, the viral vector and the non-viral vector. Viral vector, we will see with some limitation, and all the other one for all the non-viral vector are trying to overcome certain limitation. Here you reported in the upper part of the slide some example: lipid-based nanoparticle, cationic polymer, and inorganic material. And in more detail, in the non-viral vector, there is a special category as I highlight, natural material, in particular sugar, that will be the focus of my presentation. As I mentioned, viral vector, viral vector has some limitation, very well known, high immunogenicity, pre-existing immunity. For example, if in a vaccine or in a formulation, you use the virus who the patient or is in contact with before, you can practically nullify the outcome of the formulation. They have limited cargo capacity, and the most important is safety concern because of potential viral replication and insertion mutagenesis, and not last but not least, the high cost. And non-viral vectors have been developed and studied for overcome this limitation. They are much more better biocompatible. They don't have problem of safety concerning viral replication, lower risk of pre-existing immunity, and lower immunogenicity. They have greater versatility. Here I reported some example, and we will see in detail some of them. However, non-viral vectors have also some disadvantages. They have low transfection efficiency. Of course, when you are dealing with a new entity, always be careful about toxicity and immunogenicity, and shorter duration of gene expression, and sometimes poor in vivo stability. So, concerning the natural material sugar, non-viral vector as a subcategory, why cyclodextrin? Why this subcategory of natural material? I give it a long thought about how to convince the audience that cyclodextrin are the best material for gene delivery system. And my first thought was to say, okay, cyclodextrin are safe. But this is not the question, the answer that I came up. In my opinion, cyclodextrin are the most important substances for gene delivery system because they are versatile. You cannot, I'm sure that you can imagine how much is the literature about nanomaterial and formulation containing cyclodextrin, especially the simple cyclodextrin, the monomeric cyclodextrin. It's impressive. They can be included in any kind of formulation as additive, as a cryoprotectant, whatever it comes to your mind. So the versatility is the key point of cyclodextrin in gene delivery system, in my opinion. Nevertheless, cyclodextrin, you can find in several papers and patent also that cyclodextrin are used in gene delivery system because they are safe. And indeed, the native cyclodextrin are safe, are super safe compound because are naturally product derived from starch, as you know. And the cyclodextrin that have been approved and used as excipient during the years, like the hydroxypropyl or the sulfobutyl, have a very safe profile. But in the moment that you modify the cyclodextrin, you introduce synthetic modification. Here I report some beautiful molecule as an example from the literature. You have to be careful to consider them safe because you don't know the real toxicity or immunogenicity of this new compound. However, in my opinion, there are strategies that should be taken into consideration from the very beginning, from the design of the system. Here I reported a very nice article that suggested or give indication which kind of, for example, of chemical bond I use to develop a very useful excipient, like ester bond, amide, or carbonate, urea or thiourea. And in the case of cationic compound, there is a wide selection of polar head of cationic group that are safe because they are already being used in other formulation, in other drug. Same story for the hydrophobic tail. So the message here is that if you want to be preventive concerning toxicity and immunogenicity, you have to start to think from the very beginning, from the design of the molecule, and in order not to try to minimize the risk. So this was a very brief introduction, and now we go into the core of the presentation and into the core of the presentation. And here I selected some example how to use a cyclodextrin in gene therapy. Cyclodextrin as formulation and answer or additive, the literature is so wide and so vast that it's difficult to navigate it through. And cationic polymer is a specific category, and we will see go through. Polyrotaxane is an even more specific category for vehicle for gene, for nucleic acid. And the cyclodextrin uses as vector, and we will see some example of supramolecular assembly. So switching to cationic polymer. Okay, in this slide you can see the very first example of cationic polymer on the left of the slide is a tribute, let's say, to Mark Davis and his group, who was pioneering in this category of gene delivery system. In particular, the group of Mark Davis developed a very large variety of linear polymer, and they did a beautiful study about the structure and relational activity. They study everything about this type of polymer, how the space should be, what should be the cationic group that is inserted, the molecular weight, the toxicity profile. There are a very large variety and a large amount of article and patent about this one. The article that I selected, it was one of the most, one of the most interesting in my opinion, because it pointed out some key fundamental aspects. At that time, Mark Davis wanted to answer to the question of the proton sponge effect. What is? The proton sponge effect is essentially when CD-plexis or polyplexis, the complex between a cyclodextrin polymer and nucleic acid, is uptaken into the cell, is inside an endosome, and the escape from the endosome is thought to be promoted by proton efflux inward to the endosome, because if the polymer bear buffering moiety, like in this case imidazole, sucks inside a proton, and the proton will cause influx of water and swollen the endosome, and eventually rupture the endosome and release in the cytosol the nucleic acid and the vehicle as well. So in this beautiful article, Mark Davis clarified through a battery of technique that yes, the imidazole modified polymer undergoes proton sponge effect, and it escapes the endosome thanks to this one, but it is not the only mechanism at the base of this releasing. And the rigorosity of the work impressed me. That's why I selected with all the technique that clarify this point, and concluding that small modification to the molecule can cause a really dramatic change in biologic environment, which is difficult to predict. So this one was a pivotal point at that time, and for the future, that modification on the vehicle and on the property of the vehicle on the biological environment are difficult to predict. Nevertheless, it was explained and clarified that this kind of polymer having buffering effect are really able to enhance the release from the endosome. So to sum up, cationic polymer, the use of cationic polymer are highly customizable both inside and the type of genetic material. You can create polymer, both linear, both hyper branch, with different side, and encapsulate complex any kind of genetic material, both DNA and any type of RNA. This one gives versatility to the vehicle. The polyplexis, practically the complex between the cationic polymer and the nucleic acid, hold and protect the genetic material during the delivery very tightly, so they are protecting very efficiently. They can also, this kind of polymer they can be engineered to have long shelf life. This is a very important point, because we know if you can store a formulation longer without needing to extremely low temperature is always an advantage. And there are already systems that have been studied taking in account this concern. The proton sponge effect, what I discussed is already, it's very useful for this kind of polymer, and should be included at the time of the design. And one important thing is this vehicle, this cationic cyclodextrin-based polymer are useful for combination therapy. Cyclodextrin, like no one, in this case, like no other material, are extremely useful for combination therapy. I didn't bring an example for lack of time, but you can think that the cavity in this type of vehicle are most of the time unused. So you can think to include other drugs together and achieving a synergistic effect. Nevertheless, there are of course concern drawback that constantly should be overcome: low uptake and efficiency, Toxicity and immunogenicity is always a problem when you are dealing with new structure, no matter how you design the linker or the appendages to modify, it should be always taken in consideration. And in this case, the synthesis is challenging. It's beautiful. As a synthetic chemist, I say that, okay, the synthesis behind is very beautiful, is very elegant, but it's challenging. And this one is come with cost. As a second example, we switch to polyrotaxane. This one are even more sophisticated structure. And here I report an example from Professor Lee. You can see the cartoon representation on the right and the real synthetic work that they did to build up this amazing structure. Rotaxane, first of all, is a construct built up from a linear polymer, as you can see. So normally it's a pegylated polymer, and cyclodextrin enter the linear polymer, are threaded on it. Then you add a stopper in order to let the cyclodextrin slide along the polymer, but not to exit at will. And then they specifically modify the cyclodextrin with cationic group. The main idea was in this article to build up a system that is more efficient than the PEI. The PEI is a polyethylene immunopolymer, which is a golden reference standard for this kind of construct. Here you can see the synthesis very, very briefly. You have a 3-block polymer, then they put an amine on the side and tertiary amine on the side. They thread the cyclodextrin on. They put a very bulky group on the side to stop the exit of the cyclodextrin, and then specifically they modify the cyclodextrin. The synthesis is beautiful. They demonstrate clearly that the vector should show very good DNA binding ability, low toxicity, and Ig transfection for this one. They included also a battery of technique to characterize the construct. Here you can see some DNA retardation assay that show clearly that, okay, the DNA is a pick up from the co-structure depending on the nitrogen phosphor ratio at this level. And the comparison with the PEI in the case of particle size, you can see that around MP ratio around 10, you have a reaching of plateau of the different rotaxane, and also the physicochemical characterization about the zeta potential, that classically switching from negative to positive, reaching a plateau. It was then in the same article they elucidated the transfection efficiency of the complex, which is comparable to the PEI. This is NDA, the naked DNA. The performance of the rotaxane is excellent compared to the PEI. And furthermore, what is more important, in my opinion, is cytotoxicity, tested in 2 different cell lines. You can see that, okay, the rotaxane are less toxic, much less toxic than the PEI in this cell line, and also in a second, much more sensitive cell line, the availability and the cytotoxicity is more favorable. To conclude about the rotaxane and about the polyrotaxane, these compounds are very interesting and unique because are unique structure obtained from cyclodextrin. You can achieve improved transfection, reduce cytotoxicity. The other important thing is that you have reactive release of cyclodextrin and the cargo. Practically during the administration, you can trigger with some stimulus and release the stopper and burst the location, the situ that you are interested in with the nucleic acid on wheel. They have excellent biomimicry because they resemble the histone and the nucleosome, and important that they have innate macrophage targeting. This is very appealing in the case of immunotherapy. And other important characteristics like control release. Polyrotaxane have been promoted and they've been proposed for last long in release in formulation. On the drawback, we still have the complexity of the synthesis that come with high cost and still limited gene load efficiency, and again, new structure. So you have to be concerned about toxicity and immunogenicity, and also limited in vivo stability. Then cyclodextrin as pure vector. In this how to say category, cyclodextrin is used as pure vector, where also as a company we are actively involved. The main role is done from amphiphilic cyclodextrin. Cyclodextrin for build up this kind of building block are ideal because they are born as two different phases, the primary phase and the secondary phase, and they are ideal for this kind of modification when you have to differentiate the part of the molecule. For example, you can create a neutral cationic, which are the most interesting for gene delivery, and anionic amphiphilic cyclodextrin. They have a very wide range of application. They can, beside the medical application, they use as solubilizer, as emulsifier, because are similar to detergent. They are tensioactive. And in the field of medicine, they've been used extensively. There is a lot of work, tremendous lot of work, as this compound as drug delivery, in gene delivery, tissue engineering, wound healing, and diagnostic imaging as well. As an example, I reported in this category two essential examples I pick up from the literature from the group that, in my opinion, contribute more to this kind of vehicle. The first group is from Sevilla, from Carmes Ortiz Mellet and Jose Garcia Fernandez. They have been working since decade about the development of cationic amphiphilic cyclodextrin for gene delivery, and they're still doing a tremendous contribution to this field. Here I pick up an example where they were trying to build up amphiphilic cationic cyclodextrin, as you can see on the upper part of the slide, and with the targeting unit. So the idea was to transfect hepatocyte with targeting unit with the galactose targeting unit. It is known that hepatocyte overexpress the asialoglycoprotein receptor. So they were hoping to go through the hepatocyte in a specific way and to improve the transfection through this targeting unit, the addition of this targeting unit. As a comparison, they built up a library of cyclodextrin cationic, also with PEGylated, and PEGylated with the targeting unit as well. They did this one because the pegylation normally reduces the capacity of a transfection, but they demonstrate clearly through a very beautiful work that by adding the galactose unit, the capacity of a transfection was restored. Here you can see in the second part of the slide a cartoon representation of how the DNA and RNA, because they study both kind of nucleic acid, can be compacted from the structure. And indeed, as always in this group, the synthetic work is, in my opinion, amazing and fantastic, because probably I'm not objecting as synthetic chemist, but the more important is that the synthetic work is always reliable. This one I wanted to stress it as a experience in firsthand. The synthetic work is always reliable from this group. Here on the left, you can see the template, the cyclodextrin that they have been using for the modification, beautiful cationic cyclodextrin, amphiphilic with the hexanoic part on the secondary side. The same cyclodextrin with the galactose, and the cyclodextrin with the PEG, with the PEGylated part, and simultaneously galactose and PEG. The idea about the synthesis that I want to stress, because in this article, probably for the first time, they homogenize the synthetic work. The modification of and the formation of the tree antenna has been done in the same way through the thioureido approach, how we can call it. Each piece of component, they link the part together. This is a very clever and very beautiful approach, because they minimize the use of different reagents. The impurity profile is simplified remarkably, and of course the cost is reduced in this case. And after that, the study go on with the attempt to transfect pDNA and form particularly the so-called CD-plexes. They were trying to vehicle inside hepatocyte plasmid DNA, but there was a lot of frustration, because despite we will see that, okay, they could uptake and release the DNA in the cytosol, there was no expression. There was no expression of the DNA. And they were asking, they were starting to ask why DNA cannot be effectively expressed, despite we have good information about the translocation of the cytosol and the uptake. So they, in a very rigorous way, again, they analyze each step of the journey of the CD-plexes inside the cell. They clearly clarify that the pDNA CD-plexes was uptaken in the cell through confocal microscopy and flow cytometry, as you can see here on the right part of the slide. Not only, with a very sophisticated method by photochemical internalization, they proved that the release from the endosome was occurring, so that one was not the problem. And they realized, they conclude that the problem for the missing expression of the DNA was the translocation to the nuclei. So the DNA couldn't go through the nuclei. There were some barriers that they didn't allow to complete the transfection. For doing this one, for proving this one, they study also mRNA CD-plexy with the same molecule, with the same molecule, and they proved clearly that indeed the problem was the missing translocation inside the nuclei, because they had excellent expression of mRNA, selected mRNA, compared, for example, to the PEI, remarkably better transfection and transfection. Furthermore, in this experiment, they also proved that endocytosis was mediated by the receptor that they were targeting, by selectively inhibiting in a competitive manner the asialo receptor, glycoprotein receptor. Furthermore, they prove also that the viability was improved compared to for example, the PEI was still very good viability, and they could also repristinate the capacity of the pegylated version, as I mentioned before, normally as less transfection. The capacity of transfect effective was repristinated once the galactose unit was inserted again on the structure. The second example that I'm presenting here from the use of cyclodextrin as pure vector, where they are the main component for encapsulated nucleic acids, from the group of Catriona Driscoll, and we have to mention also Raphael Darcy, so from Cork, which is the second, in my opinion, major contributor to this field for this kind of category. They, in a very recent paper, they proposed the use of oligo antisense for treating Huntington disease. Huntington disease is a genetically hereditary disease that doesn't have any kind of treatment, actually, and it's caused because the huntingtin, the mutant huntingtin, cause accumulating inside neuron and finally cause neural death. In this part of the slide, you can see that, okay, the basic idea about the use of oligoantisense, a sequence of oligonucleotide is built up in order to complement to the mRNA that codify for the mutant protein and suppressing this activity, preventing the expression and translation of this one. In this work, O'Driscoll and colleague screened 3 different cyclodextrins, as you can see in the upper part of the slide. Cationic amphiphilic cyclodextrin, 2-3 substituted with primary amine, only paired 2 substituted with again primary amine and with the secondary amine, randomly substituted on the secondary side. And the component of the formulation that they use, the idea was at the beginning to select the best cyclodextrin for complexing the antisense oligonucleotide. Then they formulated the best match with the extra component like this, the phospholipid essentially, and they connected the phospholipid with the RGV, the rabies virus glycoprotein. In this case, they tried to enhance the crossing of the blood-brain barrier at the formulation level. Of course, there is a bit of synthesis between these two, but it's not a completely bioconjugate. It's a very good approach to try to improve at a secondary stage the effect of the formulation in crossing the blood-brain barrier. They did a tremendous amount of job with the physicochemical characterization during the screening of the nanocomplexes with TEM, dynamic light scattering, of course, polydispersity, zeta potential, in order to select the best nanocomplexes between the cyclodextrin and the oligoantisense. They discriminate and clarify the cytotoxicity and the stability, really important, the stability in vitro for the nanocomplexes as well. And then they went to characterize the 3 different nanocomplexes in vitro, in cell, through flow cytometry, confocal microscopy. They clarified that the best performing cyclodextrin oligoantisense was the one that it was the pair 2-3 substituted. They clarify in vitro model of neuron also the effectivity of the nanocomplex in suppressing the expression of the antigen, of the mutant antigen. And it was revealed that CD3 ASO was quite effective. Once they selected, okay, they did also immunohistochemistry study to prove again, and they practically to prove again that, okay, the CD3 ASO, you can see less fluorescent compared to the control. If there is less fluorescent in this case, the formulation is the best. And they prove once more that, okay, the inhibition of the expression was best for CD1 and CD3. Once they selected the best formulation, the best system cyclodextrin for oligoantisense, they reformulated with the RVG, in order to formulate the system to cross the blood-brain barrier very effectively. Remarkably, they developed an in-house model for blood-brain barrier. They show that this model is really rather realistic because it's very difficult to permeate, but still, with this combination at formulative level, including the RVGs, was effective in crossing the double layer, the co-culture of the two layer of cell, compared to the monolayer for the monoculture with the neuron, where there was less, how to say, selectivity in this case. So they restudy again the best, the formulation with the LG for all, again, for this kind of physicochemical characterization and in vitro characterization. And once they were satisfied for the tremendous amount of work, they switched to human fibroblasts. This is okay, it's very remarkable to arrive at this level for a formulation, in my opinion. And they proved that on human fibroblasts from Huntington disease patient, the system worked. So the system worked until a certain extent. And this is extremely remarkable because there is no therapy. It also was to remember that there was 3 huge studies that were recently abandoned because of lack of activity of the proposed system. And I think, in my opinion, this recent work can shine light for this kind of disease. To make a conclusion about the cyclodextrin, the use of cyclodextrin as a pure vector. And here you can see another example for cyclodextrin bearing targeting unit. In this case is folic acid for cancer treatment. They have enhanced cellular uptake. They are very versatile construct because you can, for example, add a targeting unit, which is very important to increase the selectivity. They have potential for control release, good stability, and protection of genetic material. And as I mentioned, high structural versatility. Drawback is still complexity of the synthesis, but the group that are involved are trying to nowadays still simplify as much as possible the synthesis and make it easier. Of course, there are problems of batch-to-batch variability, high cost, and again, this one is always a leitmotif when you're dealing with new molecule. Toxicity and immunogenicity are always concerned. And also limited clinical translation. However, nowadays, this one is changing, this point is changing remarkably. As a last example, I mean, in this, I want to present one example of the use of cyclodextrin as a supramolecular assembly. And the article that I'm presenting is for the group of Matthias Sologub, who did an excellent work with this unique work about the formation of pseudopolymer. And on the top part of the slide, you can see a summary of the proof of concept that it was addressed. They built up a very specific monomer cyclodextrin. They demonstrated that this cyclodextrin is able to cooperative force to assemble along DNA in a kind of pseudopolymer, how it's called, and it's able to transfect it. As usual, from the group of Matthias Sologub, the synthesis is extremely elegant, complicated, but elegant in any way to build up. And it's very remarkable, the idea that they had to minimize not only the amount of nitrogen. As you can see here, the cyclodextrin has essentially 2 nitrogen that bridge the primary side, and it has adamantane. They build up this molecule in order to avoid self-inclusion or head-to-head dimerization and sugar caused by sugar tumbling also, and in order to force to have intermolecular complexation in order to build up supramolecular polymer. The idea is fantastic and it has a limitless application, in my opinion. They did a beautiful study about the characterization of the construct. NMR study, through those, they proved that the pseudopolymer is formed in a very clear way. They also, through small angle neutral scattering, they prove also that they are, let's say, how many monomer are assembled, around 7, 8. So it's rather an oligomer. By fitting a model for cylindrical supramolecular assembly, even by testing different cyclodextrin, neutral and cationic cyclodextrin, they could build up, say, a DNA conformational state diagram. In the yellow, you can see that the DNA practically is unfolded. In the blue, practically with the DNA together with the pseudopolymer or oligomer, induce compacting of the genetic material. It's very elegant the way how they prove this one. And furthermore, they did gel retardation assay and they proved that, okay, the construct, the supramolecular assembly is very effective in complex DNA, and even more, and this is a very important point, they prove also the disassembly. Most of the authors, they focus on the assembly and somehow about the genetic material is compacted, is included in the system, but they don't focus about the releasing. The group of Mathieu, Sologub focus also on this one in a very remarkable way, and they clarify that the transfection is effective and also in a safe way. It occurs in a safe way by preserving the bioavailability of the cell. To conclude, this last approach has reduced toxicity through supramolecular assembling, excellent biomimicry, enhanced gene protection, and among the other one, this one is unique for the tropism modification strategy. What does it mean? Professor Mathieu Sologo already presented in the last Cyclodextrin symposium that he is able to assemble this construct with different conformation. And the final goal will be, as far as I know, in general for the gene delivery system, is to, depending on the construct, to deliver the genetic material to different organs, the different tropism. With this construct, this one is a potentiality, and I think that is gonna be spectacular the way it's gonna work out. On the drawback, we remind again the complexity of the synthesis is low cost. This one is unavoidable at the moment. To conclude this presentation, I want to say I hope that I convince or I show that cyclodextrin are not necessarily the most safe compound, at least for this use, but for sure are the most versatile substrate for the design of gene delivery system. Cyclodextrin-based systems are very promising in terms of toxicity and immunogenicity, at least one partially. Systems are ideal for combination therapy. I mentioned again the cationic polymer, or construct all the and protect the genetic material very effectively, like in the case of pseudo-rotaxane. Again, in the case, for example, of pseudo-rotaxane are suitable for prolonged or sustained release. And the architecture that it can build up can be engineered to have long shelf life, and the architecture are amenable, as in the last case, for tropism modification strategy. I would like to thank you for your attention.
Thanks a lot, Milo, for the nice lecture and the extensive explanation. You received a lot of questions. Some of them I'll ask right now. Probably the quicker ones, because we are a bit short of time, and leave the rest to the panel, which are, I think, have a bigger impact. So a few about the rotaxanes from the synthetic point of view. The first question: what is the reason that the cyclodextrin goes in the middle of the linear polymer?
The reason,
yeah,
the sliding, they build up in that specific example, they build up a 3-block copolymer, and the central part of the linear polymer, the linear polymer is somehow more effectively attract the cyclodextrin. So the cyclodextrin in that specific polymer, in that specific case, is forced somehow through selective interaction to stay in the middle of the polymer. Of course, they are able to sliding, but not to exit once they put the stopper. So they build up the polymer with 3 blocks in a way to force the cyclodextrin to go toward the center.
What could be the length of the polyethylene glycol linear polymer?
The length?
Yeah, the length of the polymer.
I don't know this one, the limitation of the length of the polymer, honestly speaking. I mean,
and would you know which is the best cyclodextrin to use, alpha, beta, or gamma? Is that a cavity
I mean, normally
specificity?
they're using alpha for threading the polymer, but okay, it's not the only it's not the only used.
And a different one about the system you showed for the adamantane supported drug delivery system. Is adamantane safe for in vivo use for such a system?
The safety, I mean, the safety is always a concern. Adamantane, okay, have been included. I mean, as far as I know, and there are drug adamantamine, for example, is a potential drug for Alzheimer or Parkinson, as far as I remember. So the molecule have been in clinical use or preclinical use for decades. But the toxicity must be assessed, because in this case, it's not only adamantane. In the article of Matthias Sologov, the adamantane is covalently linked to the cyclodextrin. So if the concern is about toxicity and immunogenicity, you have to test that compound itself, not part of it. Adamantane can be used. I mean, if you try the toxicity and you assess the toxicity, why not?
Okay, Milone, thank you very much. We'll continue in the panel.
Thanks to you.
With Katrina's support. And I also hope that we have Tao Peng on the line. Tao Peng is a biopharma research manager at Rocket Asia Pacific Innovation Center from Singapore, and he'll tell us more about the use of lactose HPBCD in biologic downstream process and formulation development. Tao, if you're ready, the stage is yours.
Yeah, okay. Can everyone hear me clearly? Hello, am I audible?
Yes, we can hear you and also see the slide share.
Okay, great. Okay. Good morning, good afternoon, and good evening. So today I'm going to share from Rocket about our in-house research about our hydroxypropene beta cyclodextrin application in the biopharma, particularly on the biologics downstream process, as well as in the formulation development. So as you all know, the biologics are basically produced from the living cells, and they usually have very large molecular structures and folded into very complex tertiary and quaternary structures. And this makes them inherently unstable. However, they still need to undergo various stress conditions from the manufacturing through the fill finish storage, transportation, all the way till the administration. This including the increased temperature, the shear stress, the exposure to light, or different source of stress conditions. So this makes them become unstable, and this unstable product will degrade or aggregate into bigger polymers and eventually precipitates. So this for sure will compromise the quality attributes of the product, and negatively impact the efficacy and the safety of the drug product. So by choosing of a suitable ingredient and excipient to be applied in the manufacturing process and the formulation will be enhance the stability of the biologics. And today we're going to introduce a new class of molecular, which is hydroxypropene beta cyclodextrin. I'm talking about new, is actually new in the biologics. Actually, it has been already well established in the small molecular drug. So here it's showing the usage of cyclodextrin families in the approved drugs. There are already 7 cyclodextrins, including alpha CD, beta CD, gamma CD, as well as their derivatives, including hydroxypropene beta CD, sulfa-butyl BCD, randomly substituted methyl beta CD, as well as hydroxypropene gamma CD. So they have been utilized in different drugs, in different routes, for different routes of administrations. And particularly for HPBCD, Here's a list of selected drugs that using HPBCD in their formulation, and they are products using for intravenous, intramuscular, as well as the oral dosage. And these are the upper part of the table showing the small molecule drug applications. And nowadays, there are more and more attention be put on the application in the biologics. So there are already two drugs. One is the vaccine. Another one is a viral vector-based gene therapy product, have HPBCD in their formulation. And we believe there are more under clinical trials for MAPs and the peptides, and we expect they are go well in the clinical trials. So in Rocket, we offer two types of hydroxypropene beta cyclodextrin. So hydroxy beta cyclodextrin is a derivative of beta cyclodextrin. So we use the hydroxypropene group to replace the hydroxy group on the BCD, so make them more soluble in the aqueous phase and bring other properties. So it makes them have a so basically the HPBCD have a bucket-like structure. So it has a hydrophobic inner structure and the hydrophilic exterior surface. So this property make them is a good complexation reagent to solubilize the hydrophobic drugs. And so that's why it be utilized initially in many small molecular drugs, and they are proved to be safe and well tolerated for parenteral applications. So in Rocket, we offer Kleptos HP and HPB. So the major difference is their molar substitution degrees. HP has a molar substitution of 0.9, and HPB has a value of 0.62. And both of them have very good solubility in water. And we actually have another Kleptos HPB LB grade product, which is specifically designed for China market that meets the Chinese pharmacopoeia. So this product, we offer traceable manufacturing and logistic chain, and the product is of high quality to meet the pharmacopoeia. Then it's already be applied in a few approved drugs, and so it has available tox study, and this data is available upon request. Then we are a sustainable supplier, provide different package like a fiber-free, or we can even provide the package for precision dispensation. So the usage of HPBCD in biologics actually have been undergoing for many years. There are a lot of prior arts by different groups. So for example, I listed a few literature here showing the function of HPBCD to reduce the protein aggregation or degradation from the interfacial stress, from the chemical stress, as well as thermal stress. And HPBCD is also an excellent lyoprotectant for the product lyophilization. Then it also be utilized for protein solubilization. Okay, so in Rocket, we believe HPBCD still has a lot of potential to be realized for their function in the manufacturing process and the formulation for different type of biologics. So we have been doing a lot of in-house studies for the new applications, as well as to bring the case studies that showcase the function of HPBCD. So here the picture showing a typical manufacturing process for biologics, including upstream, downstream, and formulation. So here I'm showing how HPBCD help in the ultrafiltration, difiltration, and then these products come up from here, will goes to the formulation. So in the ultrafiltration, difiltration, we're going to show how HPBCD, what's the compatibility of HPBCD into this filtration process, and how HPBCD can reduce the particle formation as well as protein aggregation during this stressful process. And in the formulation, show HPBCD's function to reduce the protein aggregation as well as particle formation. And we also will show HPBCD itself is a very stable chemical under various type of stress conditions. So in the downstream process, as you know, there are multiple type of filtration, including sterile filtration, nanofiltration, and ultrafiltration and the diafiltration. So these filters are made of different materials. And the first thing we do is just to check if HPBCD is compatible with these filters. Are they able to fully permeate this type of membranes? Then we bring a detailed case studies on the alpha-filtration, diafiltration of protein molecules and to evaluate the function of HPBCD during this process. So at the first, as you know, the typical filters used in the bioprocess, including the ultrafiltration, diafiltration, and the major type of filters used in this process is polyisosulfur as well as regenerated cellulose. And for the nanofiltration, the major material to make the membrane is the polyisosulfur. And for the sterile filtration, the major type of membrane is the PS and the PVDF. So we tested the fiability of two common stabilizers. The first is the polysorbate 20 and 80. And we also tested our kleptose HP and the HPB. So we basically filter the pure solutions through different type of membranes. Then we use HPLC to quantitate the amount of chemicals in the flow through. So here is the data. The finger here showing the filterability of polysorbate 20 and the polysorbate 80. So as you can see, compared to the unfiltered substance, there is a certain amount of retention of polysorbate, both polysorbate 20 and 80 on the membranes, especially on the PES membrane. There is a kind of about 5 to 15% of polysorbate got retained on the membrane, so that you see the loss of polysorbate in the flow through. So this actually align with a lot of reports. Polysorbate as a surfactant, and they also form the micelles, so they kind of retain on the membrane. And for the kleptose HP and the HPB, on the membrane we tested PS, PVDF, and PS 0.45 micrometer. We don't observe the retention of HPBCD on the membrane, which means the molecule can fully permeable on the membranes. So this will allow us to control the formulation, control the concentration of this excipient in the formulation before and after the filtration process. Then we also tested the fiability on the ultrafiltration, diafiltration membrane. And so as I just mentioned, the major membrane to make UFDF membrane is the PES as well as the regenerated cellulose. So basically we put a certain amount, certain concentration of HPBCD on the tube, then we centrifuge. Then we compare the concentration in the retentate and the permeate. For the regenerated cellulose, we can observe that after centrifugation, the concentration in the retentate apparently increased, and that there is a decrease of a concentration in the permeate, which means the HPBCD cannot freely pass through the regenerated cellulose membrane. But for the PES membrane, it's similar to the sterile filtration. It's a fully permeable, so you have the same concentration before and after the centrifugation process. So then we further tested it on the tangential flow filtration setup for the diafiltration setup and the ultrafiltration setup. So basically for the diafiltration, we keep the constant volume in the reservoir, then the volume in the permeate will increase. Then we applied 50 millimolar of study material, and we tested the concentration in the reservoir and in the permeate, and it's maintained at 50 millimolar. So the same result we obtained for the ultrafiltration. The concentration in the reservoir, in the permeate, and in the retentate have maintained at 50 millimolar throughout the process. So now we are confident to say HPPCD is fully permeable on the PES membrane. So this motivates us to apply the study on the actual proteins. So as you know, UFDF is an essential process in almost all the biological manufacturing process. So you can achieve a targeted protein concentration as well as the targeted buffers. So it applied in monoclonal antibodies, in viral vector production, as well as in the vaccine productions. But however, this continuous circulation on the membrane definitely will bring strong shear stress to the product, and the product will continuously absorb and desorb from the membrane. So these are actually quite harsh to the biologics, then which lead to the protein aggregation. So this will definitely will decrease your product yield, and the aggregated product will clog the membrane. So that causes the fouling of the filtration process. And you will expect the higher visible subvisible particles in your final product because of the aggregation happens. So in our experiment, we use two model proteins, human plasma IgG, as well as adalimumab. Then they are subjected to ultrafiltration and the diafiltration respectively, using HPPCD as a stabilizer in the process. Then for the analysis part, we use the turbidity meter to measure the turbidity, use DLS to understand the particle distribution. Then we also use the size exclusion chromatography, HPLC, to understand the aggregation of protein as well as protein recovery. So here come to the summary of the data. So for human plasma IgG, in the diafiltration process, you can see without adding HPB, and the turbidity increased from like a less than 1 to more than 12 after 4 hours of circulation. For the samples with addition of HPPCD, no matter it's 10, 25, or 50 millimolar, the turbidity is reduced by more than half after 4 hours circulation. Then the same observed for the ultrafiltration. The ultrafiltration, because you concentrate the sample and the condition is considered much harsher than the diafiltration. So the turbidity increased to more than 30, and by addition of kleptos HPB at 25 millimolar, then the successfully reduced the turbidity by half. Then for the recovery of the protein, without addition of HPPCD, there is about 10% of protein loss. Then with the addition of 3 concentration of HPB, we almost recovered 100% of the proteins. Then similar result were obtained for adalimumab as a model protein. In the diafiltration setting, the turbidity increased to about 8. In addition of 25 millimolar kleptos HPB, the turbidity maintained around less than 2. Because adalimumab is a stable protein, so it's maintained the integrity very well. We don't observe the protein loss in both of the sample with and without HPPCD. So in overall, kleptos can be applied with absolute permeability to the PES and the PVDF membranes, which suggests compatibility with filtration steps in the downstream process. This including nanofiltration, stereofiltration, and ultrafiltration and diafiltration. And as you know, UFDF is essential step, and which means you cannot avoid this step. But this step do bring damage to your product, either on the protein loss or increase the particle formations. Then for our case studies, kleptos HPBCD can enhance protein recovery from UFDF by reducing the protein aggregation. Then it can also reduce the visible and the subvisible particle formation during this process. Okay, then move on. The product we obtained from UFDF will go to the formulation part. And so here we bring some case study on the formulation, showing how HPBCD stabilized the protein during under various stress conditions for the storage, for the shipment, etc. Then we try to answer these three questions. Does kleptos HPBCD stabilize the protein in the formulation? And does kleptos HPBCD have a stable chemical structure? And how does kleptos HPBCD stabilize formulation? And we try to do some mechanistic understandings. So for biologics, especially for the proteins, they will undergo aggregation, and then aggregation can happen in basically two different pathways. They can be partially unfolded, then form a cluster that eventually become aggregates, or some molecules can be non-covalently formed oligomer. Then eventually, if there is no facilitation from the excipient, then they will form irreversible aggregate as well. So these stress conditions, including temperature, repeated freeze thaw, mechanical stress, exposure to light and oxidative agent, or the change of pH, something like that. So this aggregated product for sure is a concern for this from the safety point of view. It can reduce the drug efficacy as well. So here we are trying to show HPBCD as a stabilizer excipient in the protein formulation. So I'm just to summarize the result from our case study. And actually, the experiment is two parts. The first, we're trying to show the pure kleptos HPBCD and as well as polysorbate. What is their stability under the stress conditions? And in the second part, we subject this kleptos HPBCD and the polysorbate into the protein formulations or model protein, including bevacizumab and the adalimumab. Then this formulation is subject to stress conditions, and then we use size exclusion to understand the size profile, and we use different techniques like ITC, DLS, try to understand why this excipient can stabilize the protein molecules. Then we also use the microflow imaging system to count the particles in the formulation. So here is the result we have. At first, we show the stability of these two typical chemicals in the formulation. I will just briefly show the result because later my colleague, Dr. Zhang Hailong, will present this result in details. That would be an interesting part. Anyway, in short, HPBCD and the polysorbate are subject to heat stress in this figure. And as you can see, starting from the first week, we observed the degradation of polysorbate 20 and the polysorbate 80. And at the end of 24 weeks heat stress, there is more than 90% of a degradation happens for polysorbates. And on the other hand, the HPBCD maintain their stability very well, even after 24 weeks heat stress at 40 degrees. Then we select this sample and subject to LC mass analysis. So then we can understand the molecular profiling of these molecules. So you can see for the polysorbate 80, there is an increase of iron esterified and the monoesters, then the decrease of diesters and the triesters, which means the hydrolysis is really happens. So that makes the decrease of diester and the triester component. Then on the other hand, for the kleptos HPB, for the different for the different component with different degree of substitutions, along the 24 weeks of heat stress, you can see the consistent distribution of these molecules, which means they maintain their integrity very well under the heat stress. And the right finger showing under the other stress conditions, including the peroxide, photostability, as well as a very harsh heat stress, which is autoclave. What is their stability? So you can see the maintain of integrity of kleptos HPBCD and the decrease of protein recovery for polysorbate 20 and the polysorbate 80. Then let's look at the protein stability in different formulations. The first, as a bevacizumab as a model protein, the first finger showing during the preparation of the sample, we observed something interesting. For the protein has an initial aggregate level of 2.5%. Then we do a buffer exchange. Then we observe the decrease of aggregate level. So we then we go back to check the reference. Then found that bevacizumab is tend to form reversible aggregates. So if you dilute or you're adding some excipient like HPBCD, it can dissociate these reversible aggregates into the monomers. Then we concentrate this molecule into a higher concentration. Then no surprise, we observe the increase of aggregates after ultrafiltration process. Then also adding HPBCD, both HP and HPB can reduce the aggregation formation. Then this formulation is subjected to stress conditions like agitation. We shake at 2000 RPM for up to 48 hours, or we put at 40 degrees for 4 weeks. Then you can observe the formulation without adding polysorbate or HPBCD has an increase of aggregate to more than 2%. Then the addition of kleptos HPB or HP has a lower level of aggregate. Then this phenomenon is more obvious under the heat stress. So for the commercial formulation as well as buffer only, after 4 weeks of 40 degree stress, there is an increase of aggregate to 7%. Then in the presence of HPBCD, the aggregate level is lower, it's about 4%. So bevacizumab case study shows that HPBCD is really a good stabilizer for protein formulation. Then we further tested on the adalimumab. So this part will also be elaborated by Dr. Hailong later, but I just showing a typical data. And at the first, the left figure showing under heat stress, and the protein stability under heat stress, photo stress, as well as steroid stress. So you can observe the adalimumab is very sensitive to light. And after a one photo stress cycle, there is an increase of aggregate to 8%. Then for the formulation containing polysorbates, there is still a significant increase of aggregates. But for the sample with HPB alone or HPBCD in combination with polysorbates, it maintained aggregation level similar to T0 sample. Then also for the subvisible particle count, at the T0, you can see the small insert here. The particle number is maintained at 1,000 particle per milliliter. Then after the stress condition, we apply the agitation stress, then the particle increased to millions. for the sample without adding HPBCD or polysorbate. Then the number increase should even higher after 2 hours of agitation. But you can surprisingly, you can observe for the sample with HPBCD as well as with polysorbate, the particle number is much lesser. And the best result come from the formulation with both HPBCD and the polysorbate, which means polysorbate and HPBCD really doing an excellent job here to reduce the particle formation. Then we further tested the function of why HPBCD can stabilize the protein formulations. And at first, we tested the surface activity. We know HPBCD have a weak surface activity compared to the polysorbate. So you can see here the orange, the yellow and the gray dots showing the surface activity of polysorbate 20 and 80. And HPBCD at the same time showing a certain level of surface activity, but is much weaker compared to polysorbate. Then does it interact with a protein so that can protect the protein from the attack of other molecules? Because there is a hypothesis cyclodextrin can mask the hydrophobic part of the proteins, so that can reduce the interaction between the hydrophobic parts. Thus reduce the protein aggregation. But sometimes there's a concern the active part of the protein might also be blocked if HPBCD bind to the protein. So here we apply isothermal titration calorimetry to understand the interaction between HPBCD and the target proteins. Then we found that actually there is no interaction between HPBCD and the model proteins. So the figure here showing the curve at above is the titration, the heat titration of HPBCD into the buffer, and the lower figure showing the titration of HPBCD into the bevacizumab formulation. Both of them have a similar heat signal, which means there is no binding event happens when we mix two molecules together. Yeah. Then we also use the dynamic light scattering to measure the KD of the proteins in the presence of HPBCD. So different concentration of HPBCD added into the protein formulation, then we measure their impact on the hydrodynamic diameter of the proteins. Then we, based on this data, we can calculate the KD value. Then the data tells in the higher concentration of HPBCD, the interaction between proteins are reduced. So there is a repulsive force generated in the presence of HPBCD. So with all the data, we conclude that for the protection mechanism, we don't think there is a complexation happen between HPBCD and the protein molecules. However, the weak surface activity as well as the preferential exclusion of HPBCD on the target protein is the reason why HPBCD can stabilize the proteins. And from the protein case study, we know that HPBCD is effective in reducing the stress-induced protein aggregation, and then it can also reduce the particle formation under stress conditions. And in addition, HPBCD is a very stable chemicals, especially comparing to polysorbate, which always have a stability concern when it's subjected to the biologics formulations. So last but not least, I want to use these slides to summarize our findings of HPBCD application in the manufacturing process. So the UFDF is an essential step for manufacturing process, but it's a harsh process for the biologics. So HPBCD at first are fully permeable on the PES and the PVDF membrane, so which means suitable for application in the sterile filtration, nanofiltration, and ultrafiltration and diafiltration. So this can control your excipient concentration in the final formulation. And during this process, it can effectively reduce the protein aggregation and the formation of visible and subviscous particles. Then for what bring forward into the formulation, the HPPCD can continue to play the stabilization function. It is a superior, it is an excipient with a superior stability comparing to polysorbates. Then HPPCD can effectively reduce the stress-induced particle formation and the protein aggregation in various stress conditions. So we can expect the shelf life of the protein will be extended. Then with regards to the protection mechanism, we assume it comes from the weak surface activity and the preferential exclusion. So it can reduce the protein-protein interaction. So with this is the end of my presentation. Thank you for your attention.
Thank you very much, Tao, also for keeping your time and delivering this very informative lecture. We have, again, quite a few questions. Some are more related to general considerations and regulatory aspects, so I'll leave those for the panel, but there are a few which are more practical. Several people asked about the conditions of the heat stress studies. Could you tell us about those? What was the temperature?
Okay, so the model protein we choose is the monoclonal antibodies. So basically they are stable molecules. So the condition we use is actually the 40 °C, and we apply the 40 ° for up to weeks. So we're sampling at like a 1 week, 2 week, up to 4 weeks of time.
Great, thank you. Another one is about drawing direct comparisons. So with the immunoglobulin G and adalimumab case studies that you have shown, have you compared the effect of kleptos or the performance of kleptos to polysorbates in these cases?
It's a good question, and we didn't apply polysorbate here because UFDF is almost a last step before the final formulation, which means you need to control your excipient concentration for your UFDF final product. For the polysorbate, you will form micelle, and also it interact with the PES membrane. So which means you cannot control the final formulation, final concentration in your final formulation. So it's basically is not the appropriate excipient to be applied here. So that's why we didn't try it. But from the literature, there's a literature showing polysorbate does a good job to reduce particle formation as well as protein aggregation. But in that paper, they didn't measure the concentration of polysorbate before and after the UFDF process.
Yes, thank you. And we have one about the concentration of cyclodextrins in the TFF process. Would you know, do you have any experience? Why is it concentrating in this part of the production?
Sorry, can you repeat the question again?
Question is, do you have experience why the cyclodextrins are concentrating in the TFF process during the production?
Oh, actually, there is the cyclodextrin is not get concentrated in the TFF process. Okay, so there are two types of TFF in the market for the manufacturing. The first one is regenerated cellulose, and HPPCD is not permeable on the regenerated cellulose, so it's not compatible with the regenerated cellulose. And the material we use here is the PES-based TFF membrane. And during the whole process, there is no the concentration of HPPCD is controlled exactly at the concentration we input. So there is no concentration or dilution of HPPCD concentration throughout the process.
Perfect. Thank you very much for the clarification. So dear attendees, now we are around the half of the webinar. So we are taking now 4-minute bio break until 1:40 Central European time. So we'll continue in about 4 or 5 minutes with the lecture of Heilong.
Okay,
Stay
thank
tuned
you.
until then. Yeah, just a few minutes. Okay, so I hope you could stretch your legs and grab a cup of coffee or whatever you need to keep yourself sharp. And let's continue with our next and last lecture before the panel, which will be delivered by Heilong Zhang, who is an analytical research manager at Rocket, and will show some nice comparative studies on polysorbates and HPBCD on stability and antibody stabilization.
Hello everyone, my name is Heilong. I'm from Rocket Pharma Applied Science. Today I'm very pleased to capture your attention. I would like to report our recent study on polysorbic and kleptos HPB, which is hydroprofile beta-cyclodextrin, to compare their stability, performance, and their benefit in biopharmaceutical formulation. I will start with the background and the scope of this study, then discuss the different behavior of polysorbic and hydroxyprofile beta-cyclodextrin individually when they encounter stressful conditions, and finally, the effect of both compounds on different protein stability in formulation are assessed. Use a case study with adalimumab antibody. Polysorbates are widely used sufficient in biopharmaceutical formulation to prevent protein from denature, aggregation, surface absorption, account more than 70% of all market antibody formulation. However, the polysorbates are chemically diverse mixture, have a big lot-to-lot variability. More importantly, polysorbates are prone to degrade by oxidation and hydroxylase. As illustrated in this figure, degradation of polysorbates produce peroxide and fat acid, and those degradants could induce protein oxidation and aggregation to form insoluble particles and negatively impacting protein quality and stability. Beta-cyclodextrin has emerged as an interesting class of a molecule. Native beta-cyclodextrin and derivatives are cyclic oligosaccharides with a box-like structure. They have been well-defined structure and are chemically stable. Hydroxyprofile substitution in beta-cyclodextrin increases its solubility. The increased HB beta-CD is a well-established excipient and used in many approved small molecule drug formulation to improve the solubility and bioavailability. And HB beta-CD has also been widely explored as a stabilizer in biological formulation in many studies. Most recently, in the mRNA vaccine from Johnson & Johnson. To answer if HB beta-CD can serve as a functional alternative to the polysorbate, we would like to share our comparison result of HB beta-CD and polysorbate with respect to their chemical stability and benefit on protein stabilization. To qualify HB beta-CD as a potential alternative, the stability of HB beta-CD in formulation needs to be assessed. In this work, we evaluate for first time the chemical stability of HB beta-CD in aqueous medium and various pharmaceutical relevant stress conditions such as thermal, lead, and oxidation stress. A direct comparison has been performed by subjecting 3 grades of HB beta-CD, polysorbate 80 and polysorbate 20 to the same stress condition. For some stress tests, some sample was placed in 40 degrees 75 RH accelerated chamber for a period of 24 weeks. Autoclave sample was subject to 5 autoclave cycles at temperature of 121 degrees. For photostability study, we follow ICH Q1B gallon, wet lead for 1.2 million luxury hours, and UV lead for 200 watt hours per square meters. Finally, we evaluate degradation due to oxidation by adding 3% of hydrogen peroxide. We first evaluate polysorbate and galactose product and heat stress. When subject to mild thermal stress at 40 degrees until 24 weeks, different grade of the HP beta CD shows a slight change in product recovery with 90.7%, 100%, and 91.4% for galactose, HP, HPB, and HPB-LB respectively. While polysorbate 20 and polysorbate 80 both suffers a significant degradation with product recovery only 11.5% and 7.3% respectively end of the study. To gain insight into degradation pathway and the degradation product, the component profile of polysorbate 20, 80, and galactose HPB and their change and thermal stress has been examined to compare the stability difference. HPLC-based separation method with combination of different detection technologies such as charge aerosol detecting and mass spectrometer has been developed to examine the chemical component of polysorbate and to monitor the change of that corresponding degradant. As shown in the left, HPLC method is able to separate, identify, and quantify the multiple ester species in polysorbate 20 and also to monitor the change of their corresponding degradant. This is an overlaid chromogram of polysorbate 28 over the time. The main peak has essentially completely degraded, and a degradant product appears at the changing time of polysorbate. Again, we examine polysorbate 80 stability by using the same strategy as shown in overlaid chromatogram. The polysorbate 80 samples degrade over time and heat stress. Major components such as diester, triester almost completely degrade after 24 weeks and heat stress. Here we develop a HPLC-based separation technology to evaluate HP beta CD molecule profiling with the detection by using charge aerosol detector and MS detector. We are able to identify 11 components in a sample of the galactose HP beta CD, and each of the substituents of cyclodextrin can be analyzed with this molecule profiling information, and each of that substituent, the stability can be assessed as well as this control and the different stress condition. For both polysorbate 20 and 80, it's clear to see the main component of ester, including monoester, polyester, decrease over time, while the unestified component increase up to compare unstressed sample. This observation is consistent with the change of overall concentration of polysorbate 20 and 80 determined previously. In contrast, polysorbate HPB has been found no obvious change for each of cyclodextrin substitute within 24 weeks, and thus exhibits great stability under the thermal stress. To further understanding the chemical stability of polysorbate and HP beta CD and a more serious condition than heat stress condition mentioned before, other force degradation studies such as light stress, oxidation, and extreme heat stress were carried out to determine the intrinsic stability of both polysorbate and HP beta CD. When subject to the autoclave for 5 cycles at 121 degrees, 85% of polysorbate and 89% of polysorbate 80 remain in a solution. When subject to the oxidation both polysorbate 20 and 80 suffer around 10% the product loss within 1 week. Similarly, exposed to the light stress condition, as described before, result in the 7 and 10% of product loss for polysorbate 20 and polysorbate 80 respectively. In contrast to the polysorbate, all HP beta CD remain stable when subject to the autoclave, light stress, and oxidation stress. Again, we examine the component of the polysorbate and HP beta CD and monitor the change in their corresponding degradant and different stress conditions. When subject to those 3 stress conditions, all major components in polysorbate 20 and polysorbate 80 decrease, while non-acidified component significantly increase compared to those in unstressed sample. For HP beta CD, the well-overlaid chromatogram of unstressed and stressed sample indicates that there's no significant degradation observed for all substituents and the autoclave, oxidative, and light stress. In summary, the chemical stability result of polysorbate and the HP beta CD product and its various stress conditions were outlined here. Those results combined with the component profiling analysis all serve to demonstrate all grades of our HP beta CD product show great stability than both polysorbate 20 and polysorbate 80 at different stress conditions. Now let's move on to a case study. In order to identify the influence of polysorbate or HP beta CD on protein stability, we choose commercially available and best-selling monoclonal antibody, adalimumab, as a model protein to evaluate HP beta CD benefit on protein stability. Adalimumab antibody is the first fully human recombinant IgG2, IgG1 anti-TNF-alpha antibody, which has been used in treatment of wide variety of inflammatory conditions. A fair number of studies show that thermal stress causes aggregation and fragmentation in adalimumab antibody, and a shear stress induces significant particle formation, and also the light stress has a negative effect on antibody stability. In this study, physical-chemical stability of adalimumab antibody in formulation with polysorbate 80 or HP beta CD as a stabilizer was investigated. As such, a series of stability testing uses different distress conditions, including mechanical, thermal, and light stress, were carried out to force the adalimumab antibody default aggregation and aggregation. Then stress-induced aggregation and aggregation profile was measured and analyzed by different analytical strategies, such as size exclusion chromatogram, cation exchange chromatogram, microfluidic imaging, and dynamic light scattering. Now let's first examine subphysical particles in different formulation and agitation stress. Within 2 hours, stirring led to a significant increase in subphysical particle formation in all formulations compared to unstressed T0 sample. The presence of either galactose, the HP beta CD, or polysorbate 80 was able to reduce the formation of a particle significantly. The aged polysorbate 80 in 2018 produced slightly higher particle compared to the new batch of the polysorbate 80 made in 2021. Due to their high profile value, then cause a great extension of the protein oxidation. Interestingly, polysorbate 80 was used with the HP beta CD, the formulation prevented the further particle formation down to 10% of the particle formed in control buffer. So when subject to heat stress and 40° 75% RH, a modest increase of particle formation in control buffer was found, and more particle over time during 4 weeks incubation. Adalimumab antibody in control buffer generate the highest number of subphysical particles. By contrast, the presence of that HPV beta CD in the formulation significantly reduce the formation of aggregation, and they offer similar protection as polysorbate 80 against the particle formation. Similarly, when subject to the light stress, the limb antibody formulation in control buffer produce the highest subphysical part among all formulations. All other 4 formulations, either containing polysorbate 80 and the HP beta CD, reduce subphysical particle formation. However, both the HP beta CD include the formulation offer a better protection by preventing more particle formation compared to other formulas. Further, the stability of adalimumab antibody in 5 formulations and a different stress condition was assessed by SEC HPLC by measuring aggregation and fragmentation in a formulation. The data shows that stirring stress did not increase aggregation and fragmentation level in all 5 formulations. Under the thermal stress, no significant difference of aggregation and fragmentation in all formulations. Only the HP beta CD containing formulation showed a slightly reduced fragmentation product compared to control buffer and the polysorbate 80 formulation. Interestingly, aggregation level decreased in all 5 samples, likely because the aggregate antibody was removed from the formulation after centrifuge before HPLC running. Comparison of all these results with the composition of unstressed antibody revealed that a negligible influence of temperature stress on number of that fragmentation detected. Then we examine the thermal stress effect at the elevated temperature at 50 degrees to evaluate antibody stability in 5 formulations. And the thermal stress HP beta CD containing formulations present a significant lower aggregation level compared to polysorbate formulation. HP beta CD containing formulation also show a lower fragmentation level and a higher monomer recovery compared to polysorbate 80 formulation. Lastly, the photo stability of adalimumab in 5 formulations after exposure to ICH lightning condition has been investigated. Light stress led to significant protein aggregation in control buffer and polysorbate 80 containing buffers. 2 collaptors, the HP beta CD formulations present a relatively lower protein aggregation compared to other formulations. Similar result was observed for antibody fragmentation level, in which the HPV beta CD formulation should lower the fragmentation level in all formulations. In terms of monomer, both the antibody sample in HP beta CD formulation gain a better recovery than other 3 formulations with around 98% recovery. Also, we examine the charge variation of the antibody and the lattice stress. We have observed a significant shift in relative abundance of charge species and the thermal stress. Galactose HP beta CD containing formulation gain most high recovery of the main charge variant amount in 5 formulations. Our data also shows that the main species disease predominantly while both acid and basic variant increase and the light stress. The presence of the HP beta CD in the formulation efficiently prevents the loss of mean species, and suspect, suppress the basic variant enhancement. In summary, quantitative analysis and profiling characterization has shown that HP beta CD product exhibited greater stability than polysorbate and for stress condition, heat, elevated temperature, oxidative and light stress. In the formulation study, present of either HP beta CD or polysorbate AD was able to reduce formation of subphysical particles significantly in the stirring stress. Exposure to light stress induced the aggregation and fragmentation of adalimumab antibody in control buffer. HP beta CD present a better protection from the light stress by reducing the protein aggregation and fragmentation level, as well as enhance the charge variant stability than polysorbate AD formulation and a control buffer. So we believe Galactose HP beta CD has surface activity and may serve as a functional alternative to polysorbate in the biopharmaceutical formulation in the future. So we would like to thank you very much for your attention. Thank you.
Before we go with the panel, I'd like to ask all the experts who are here participating to switch on the cameras so that we can see everyone. Okay. Okay, that's nice. So I see that Triona and Carmen are also here. So I'll continue with some introductions so that everyone is properly introduced. So in the panel, besides the speakers, we have Dr. Carmen Popescu, who is Global Pharma and Biopharma Program Coordinator at Rocket America, and who has a lot of expertise, of course, in cyclodextrins and their uses with proteins. And we have Katrina O'Driscoll from the University College Cork from Ireland. And as you could hear from Milo's lecture, Katrina has over a decade-long expertise in using cyclodextrins in gene delivery, as well as other drug delivery systems in gene delivery. And she's coordinating currently a European grant, which is focusing on creating novel drug delivery systems for these applications. So welcome, Carmen and Katrina. Please check if we can also hear you.
Thanks, Thomas. Yeah, I can hear everybody.
Yeah. Hi, Katrina. Hi, Carmen. So we have quite a few questions that we can discuss in this session. But if any of you have anything to add to the questions, I encourage you to raise hands. There is the possibility, and just in order to wait for everyone's feedback. So we have a few general questions, and we could maybe start with those as those are the ones that affect both topics, both the gene delivery and the protein formulation topics. Alan, we have received a few questions about the general safety and toxicity related to the cyclodextrins, especially concerning the kidney toxicity, and that EMA has a guideline on that, or some kind of official standpoint on that. Could you elaborate what is the current status and regulatory aspect of accepted cyclodextrins in the market?
Yes, sure, Tamas. I assume that the question is maybe linked to the guideline in EMEA. What should be the notice in the excipient, in the package, the leaflet package of a drug product? What should be mentioned about excipient? So this is common to, I would say, all excipient, at least those for which we know they can have an effect for some particle specific population. So this has been done to, I would say, almost all excipient. And then just to give you the history of this is France who started with this, with a generic of small molecule. When a patient was taking the originator that do not contain, like, for instance, lactose as an excipient, while the generic contain lactose, for some patients who were intolerant to lactose, they had some effect. So coming to cyclodextrin, I really encourage all people to go on the EMEA website, because they have done a great job in 2017. It's a kind of a document called Q&A to describe all, I would say, the safety of cyclodextrin and modified cyclodextrin, which are in approved drug product in the Europe. And they have done it by route of administration, oral, parenteral, topical, and so on. And they base this on the safety data, all the tox data they have in the marketing authorization files. So saying that, coming back to the leaflet, once they have done this Q&A in 2017, they have updated the guideline about excipient and the package leaflet. And when it comes to cyclodextrin, they take it as it is in the list, either which route of administration, either parenteral or oral. And what has come, it's first to be careful, they put, so the information on the leaflet, be careful, it's we don't have sufficient safety data for children, so do not use it. And this is for any cyclodextrin, don't use it for children below 2 years. And also it is well known, like the native cyclodextrin, they might have a renal toxicity. This is really well known. So be careful for the patient, there is a mention, who have some renal deficiency. So this means, or does not mean, that cyclodextrin are not safe. It is only to put some precaution of use for some category of population. Tamas? We lost you, Tamas?
So thanks a lot. So let's get a little bit topic specific, because the questions are not easy to that they're not fitting both topics. So let's start with the gene delivery first, and I'd like to hear Kite Fiona's views on one of the aspects, because apparently you have the most experience here. Someone was asking a question: what is the vector capacity of a known pharmaceutical excipient like a hydroxypropyl beta cyclodextrin versus complex substituted amphiphilic or cationic cyclodextrins? Have you done any any such studies in your past? You're muted, Catriona.
Sorry, thanks, Thomas. You're asking about, or the person is asking about the capacity of non-amphiphilic versus amphiphilic cyclodextrins as delivery vectors. Is that correct?
Yes.
Yeah.
Well, not necessarily non-amphiphilic, like amphiphilic cyclodextrins or cyclodextrins designed for this purpose compared to common excipients like a hydroxypropyl beta cyclodextrin, if that would also be suitable for the purpose.
Yeah. Well, I think we need to first of all look at a list of criteria that we think that should be in the cyclodextrin in order to deliver the nucleic acid. So the nucleic acid is a very large molecule. It's negatively charged and hydrophilic. So it doesn't go across membranes very easily. So in order to try to chaperone it across the membranes, we've tackled it by synthesizing cationic materials, which tackle the excessive or neutralize the excessive negative charge. And then we also have the amphiphilic cyclodextrins that can self-assemble around the nucleic acid. And the presence of the lipid component also enhances membrane permeability. So I think from our experience, those are pretty essential criteria from a physicochemical point of view that have to be built into the cyclodextrin in order to get over the barriers, the physicochemical barriers associated with the delivery of RNA, any nucleic acid, actually. Does that answer the question?
Yes, of course. I think it does pretty much that we need custom cyclodextrins here, because the functional properties related to the purpose are completely different from what we can achieve with common ones. Milo, you have shown us different synthetic strategies for gene delivery and shown us the advantages and the handicaps of each. From a chemistry point of view, which one of you think has the best potential to become an actual drug delivery system?
Well, thanks for the question, Thomas. I think that from every cyclodextrin could be potential suitable for become an effective delivery system. Then, okay, if my favorite, I mean, because I put inside my personal point of view, is the use of cyclodextrin as pure vector. It has the larger potential. But I wouldn't be completely honest, because the idea to use cyclodextrin as pseudopolymer and the possibility to target different organ depending on the conformation is really appealing, and I really love this potential. If you remember in my presentation, the last example from the group of Sologol, that is very, very appealing, and I think there are pharma companies that actually are heavily working on this one to target the organ depending on the conformation and configuration of the CD plexes. So I don't know if I answered your question.
I also meant if all of these strategies are in your expertise, scalable, probably acceptable by regulatories.
I have been working with most of the example that I presented. Beside polyrotaxane, I never had the luxury and time to work with them. So about that one, I'm a bit less prepared from a practical point of view. For the other system, yeah, the upscalability is not an issue. I mean, if the synthesis is studied from the beginning and designed in a way to be upscalable, it's possible. There is no problem. There is just need of time to be really careful in the design, assess the toxicity and the immunogenicity from the beginning, as I was mentioned. But there are no practical limit, in my opinion, for the large-scale production of this construct, both cationic or pure vector, cationic, amphiphilic, or pseudopolymer.
That is good to hear. Okay, Katrina, we also received a question, and probably you're the best one to address it, about the the progress that has been made with these drug delivery systems. So we have seen a lot of examples on using them in transfection in various cellular models, but how advanced is this research field? How advanced are the efficacy studies, the safety studies with respect to in vivo experiments, and where are we on this development timescale?
Yeah, I think it's at a relatively early stage still. We and others have managed to produce some in vivo efficacy data. In our case, we have animal data with siRNA for a range of diseases. And we have preliminary tox data. So it's hard to kind of put an exact time scale on it. I think there's a lot of activity, so it'd be hopeful that things would progress pretty quickly in the next 4 or 5 years. I think just to pick up on one of the comments that Milo made there, we're all very familiar, I think, with the use of lipid nanoparticles to deliver these nucleic acids. And they've worked very well, obviously, in the COVID vaccine, but there's a big difference between using a delivery vector for kind of intramuscular use and comparing that to systemic administration. And some of the siRNA products that are on the market that are given by parenteral use are used to treat liver disease, and with the lipids. So I think one of the big challenges about having other non-viral vectors such as cyclodextrins is the ability to deliver to organs other than the liver. So this is what Milo was saying. By tailor making the materials, we believe that it's possible to get a better biodistribution so that they don't all end up in the liver. And that's a big challenge, I think, for a lot of the companies, and particularly for the companies that are focusing on lipid nanoparticles, because they have an endogenous, if I can call it like that, trafficking mechanism that brings them to the liver. And that's fine if it's a liver disease you're targeting, but if you're trying to get it to any other disease site, you need other materials that can be tunable to get you better targeting to other diseases and other targets or other tissues and organs within the body. And I think there's an opportunity there for cyclodextrins to achieve that.
Speaking about targeting other organs, so at GeneGut you're working on an oral gene delivery. So yeah. Is that something that's one of a kind, or there has been attempts to do that?
Well, I think that it's definitely the first time that cyclodextrins have been explored in this way. So in terms of GeneGut, which is the name of the project, we're trying to design an oral delivery system which will achieve localized delivery. So we're not looking in that case for systemic delivery. We just want it to be released at the right site in the intestine where the inflammation occurs. So I think the approach that we're taking is pretty unique because it combines a range of different biomaterials, including cyclodextrin, to package the materials. But we also, I think there are a few kind of strands of attack here. You have to have the right biomaterial, but you also need to identify the right target from a gene perspective. In other words, what genes are you going to knock down by using siRNA, or what genes are you going to promote using messenger RNA? So you have to have a good idea of the disease itself and the mechanisms of the disease. And then the last kind of attack point is to have a capsule that will get to the right site. So you don't want a capsule that delivers or breaks up in the stomach or in the upper part of the intestine. We need to have a capsule which is specifically designed to release right at the disease site. So I think when you put all those parts of the jigsaw together, that's what makes the GeneGut different to other approaches that have been used before. And I suppose I should add, we also have partners in the GeneGut consortium who are using very advanced in vitro cell culture models of the disease, where we can test our systems in vitro. And then we also have in-house expertise using pig models. So we're hoping to test our prototypes eventually in vivo in a pig model, and pigs are chosen because their physiology of the intestine is very similar to man. So I think that's a good choice there. So those are the kind of advantages of the gene gut project. I think that makes it different to other approaches that have been used in the past.
That is indeed very, very fascinating and a very, very unique idea. When considering ideas, and maybe I'll ask Milo this one first, but Kaitriona's opinion would also be interesting. So RNA delivery systems are now evaluated and used in quite a lot of different applications and models. Of course, they got very popular with all the vaccines, especially the Pfizer and Moderna vaccines recently, but eventually they are mainly most of the research is done in cancer therapy, but we have seen today other indications, neurodegenerative diseases, genetic diseases, autoimmune diseases. So do you think that cyclodextrins would be something that are generally applicable? I mean, even in potentially in mRNA vaccines, or they would rather have a specific role and a specific application or applications within this whole drug delivery field or gene delivery field?
I go first. I mean, in my opinion,
yeah, you're the first, Milo.
They can be applied in everywhere. As I was trying to stress from the talk in the presentation, the strength of cyclodextrin is versatility. If you tailor-made or you design them for doing specific tasks, they will. At least this is my hope and opinion. So they are not dedicated to a niche or to something extremely specific. They can be of general use once you tailor-made. I mean, unluckily you don't have a cyclodextrin that can do all the job. This one I think is very naive to think it would be wonderful, but it's not the case in my opinion. You have to put effort to dedicate to design and customize the cyclodextrin for that purpose. In that case, you can succeed. In this case, the versatility is the strength of this building block, like any other, in my opinion. I don't have doubt about this one.
Right, Fiona?
Yeah, I would support Milo there. I think that because of the unique structure of the cyclodextrin skeleton, there is the possibility to synthesize a library of different modified cyclodextrins. And as Milo said, once you know the disease target that you're going after, you can synthesize cyclodextrins with targeting ligands. I mean, concentrated on amphiphilicity and cationic, but you can also add targeting ligands onto the cyclodextrins, which can be chosen specifically for a disease site. So, and the other thing I think is with the cyclodextrins, and we've done a little bit of this here, is if you have a library of different functionalities, you can co-formulate them together. So you can have amphiphilicity, you can have pegylated cyclodextrins, and then you can have targeted cyclodextrins. And by combining them or co-formulating them together, you can get bespoke nanoparticles which are targeted for a particular disease application, which is, I think, what Milo is referring to. So once you know the disease target, you can definitely modify the formulation based on the cyclodextrins.
Okay, thank you both. I'll just have one last question on the gene delivery topic, unless we receive some in the Q&A. So we are all probably biased in the favor of cyclodextrins because we all love these molecules, but if you try to be objective, what is the drawback of using cyclodextrins in drug delivery against lipid nanoparticles or other alternative drug delivery systems? And you cannot say there are none.
Be objective, okay, as you mentioned, is difficult. Sorry, Katrina, you want to go first?
Yeah, I might kick off there, Milo.
Sure. Go ahead.
The challenge is to ensure that you get good loading into the formulation. So that can sometimes be a challenge, that you get an adequate dose of your nucleic acid, be it messenger RNA or siRNA. And also you need to make sure that whatever chemistry you're doing, that there's no immunogenicity produced with these molecules. So, you know, because in general, with these nucleic acids, you're going to want to administer them, you know, several times. So we have to make sure that the immunogenicity is not an issue there. So they would be the two of the things that would come to my mind to start with anyway. So Milo, maybe you want to add to that.
I just want to add, particular from the synthetic point of view, that when Thomas asked this, when it comes to my mind, cyclodextrin have a cavity, which is an enormous advantage, but from the synthetic point of view is a horror. I mean, for synthetic chemists, the cavity can be an hinder. I mean, there's something that should be overcome. So it's really, the cavity should be used, and it's the peculiarity of this microcycle, for this sugar, but it's also at least from the synthetic point of view, much more complicated. So it's a double edge, how to say.
Okay. But thank you both for the clarifications and the opinions. If there are further questions, we can get back to it. But now let's move a bit towards protein formulations. No, don't switch off your cameras. So protein formulations. Carmen, we have a very general question, and I guess that it's for you. So someone's asking about how is the effect of hydroxypropyl beta cyclodextrin explained on preventing protein aggregation? So what is the mechanism of action here? You're muted, Carmen. Sorry.
Okay, so basically the mechanism is in discovery. Okay, if I want to make a comparison, it's like DMSO is the best ideal solubilizer for everything. Okay, that's why in discovery you use DMSO and you are happy as a synthetic chemist. Okay, so I think cyclodextrin have some of the magics like DMSO in the fact that having this low surface activity is not a surfactant, but behaves like a surfactant. Secondly, that is allowing it to be preferentially excluded from the surface of the proteins. That means they do not react. So you have just a thermodynamic interaction with the protein. And in the a third aspect will be, I think, what is doing to prevent aggregation is like a shield, is not allowing the hydrophobic domain of the proteins to interact with each other. So it's like a shield, but in the same time doesn't react with the hydrophobic domain on the protein. So it's a shield protecting the interaction between two neighboring proteins. And being neutral, that is the most important thing.
Yeah, I just wanted to ask that. Okay, so but that's something that most cyclodextrins could do eventually. So
not eventually. So this hydroxypropyl beta cyclodextrin has the advantage of being neutral. So there are many aspects besides we consider here all the stressor, mechanical stressor, temperature, photo effect, and so on. But we have to look a little bit to the basics, as the fact that hydroxypropyl beta cyclodextrin is neutral, comparing with, let's say, a sulfobutyl cyclodextrin, which is carrying a negative charge. We know the proteins generally, are negatively charged. The membrane are negatively charged. DNA, mRNA, all monoclonal antibody are generally negatively charged. That's why it's very difficult to use SBE as a delivery system. This neutrality from the charge point of view of hydroxypropyl beta cyclodextrin is relevant because it's allowing just thermodynamic interaction with proteins, not a reaction, not covalent bonding. So that I think is the advantage of hydroxypropyl beta cyclodextrin.
Tao, do you have a view on that? You have done a lot of experiments here.
Yeah, I think Carmen just nicely summarized the mechanism in terms of why HPBCD and not others. I just share aspect in terms of regulatory because as Milo mentioned, he's a fan of modification on the cyclodextrins. Definitely, that's the future to explore the more potentials of different cyclodextrins and derivatives. But as a low-hanging fruit, I think, in my opinion, this cyclodextrin who are already got approved in the got applied in the approved drugs like HPBCD, HB gamma CD, and SBECD might be the the first choice for the drug developers. And among these, as Carmen just mentioned, SBECD is not suitable because of the compatibility with the large moleculars, and the HPBCD then can be, is a logical choice. And it turned out is very effective from all the publications we see and also from our own hands.
So just to capture, if I understand your combined views correctly, you think that the well-established safety profile and the very good safety and toxicology data supporting HPBCD, along with the proven functional properties, support that it should be used. It should be the first choice, at least for protein formulations. There could be other cyclodextrins that could work better or could be developed for that purpose in the future. But for now, this is the best choice if someone doesn't want to start from scratch with something that's completely unknown to the pharma.
Yeah, exactly. Yeah, I think even for HPBCD, actually is at the beginning stage for the application in the biopharma. So I think there's a lot of work for us to do to explore, to maximize the applications.
Since you said that it's in the beginning stage of biopharma, so you have shown a lot of studies supporting the applications. How far are the further studies with cyclodextrins in protein formulations?
So as you all know, biopharma drug development is a long process, usually easily more than 10 years. So I don't know how long it will take, but what I can share, I can see there are more and more interesting from the publications and even from the people I've talked to, and they are working in the biopharma industry. They have a higher interest in applying HPBCD in their lab, in the preclinical development, into the process and in the formulation. So, and I think there are already a few drugs under clinical development that have HPBCD in their formulations. So I think the good news will come in hopefully in next few years.
Okay, that's interesting. Carmen, anything to add?
If I may add, is the fact based on my interaction with biopharma companies. What I realize is the fact that in biopharma, you have a very simple type of formulation. You have a buffer, you have some stabilizer, you have this. So it's sort of standardized formulation generally. Okay, so it seems like people are just starting to hear about that. Okay, polysorbates can have some issue, not at the beginning of the formulation. You'll find out after you put on stability. So, okay, what is the solution? It started with using polyethylene glycol as a replacement, other replacements, but it seems like cyclodextrin is the best bet. But people are still in the, how can I say, information step about these good properties of cyclodextrin. So the more webinar like this and people are aware, people will start to use them. And we found some of the properties of cyclodextrin from the people who are trying it. So this specific properties of to replace polysorbates in ultrafiltration diafiltration came from a customer saying, okay, I am using cyclodextrin, I'm very happy. I don't have to change my membrane all the time. So these are facts coming in cooperation between us, and the better we can propagate this news will save time to the scientists. And many of them are just following the recipe. So let's help them to go out, okay, to think out of the box.
Yes, thanks a lot for that, because that's also something that's very interesting. And I also wanted to ask, because you interact a lot, both you and Tao and with biopharma companies. So where are you with introducing this new application? Because this is a fairly new application for cyclodextrins. How general it is for a company dealing with protein formulations, that cyclodextrins are really on the shelf, and when they start looking at formulation agent, they are actually evaluated. Is that a common practice or something entering the trends? Where are we with this? How many webinars do we need to make?
Maybe 10, 20, 30, 40, 100 until everybody will be aware. But with this kind of communication, okay, the best bet was when many people are doing high throughput screening. Now, when you do high throughput screening, they want to use low concentration of excipients as low as possible, okay, you know, the CRA, okay, less is better. And sometimes I don't know if everybody knows exactly how cyclodextrin are working. So until we can deliver the understanding of why cyclodextrin and how they behave in stabilizing and inhibiting aggregation, then in this high throughput screening is sort of general way. So I think it is necessary to talk more about the mechanism, what are cyclodextrin, why they do what they do, and so on. So it will take some time.
You stressed a lot, Tao and Daylong, the comparison between cyclodextrins and polysorbates here. And I would like to understand, maybe not necessarily from you, but Elam and Carmen, the feedback from the industry. So is the superiority in safety and comparable performance in protein stabilization enough, or there are or the economic concerns, the safety concerns, the novelty concern that it's not used that widely, overwhelming these safety advantages? What is the feedback that you're receiving?
For me, first is the stability, shelf stability of the proteins.
That's the most important.
That was a moment when, you know, because at the beginning, you cannot see polysorbates having some issues, okay? So after the stability studies, they realized there are some stability issues. Now we have to look for an alternative. Now, some people we recently started, okay, it's the last 3 years to do this kind of study. So even for Rocket is something new. And we didn't, how can I say, promote like now in a webinar until we have real solid data to prove, yes, this is a comparison, this is what you are getting, and so on. So yes, there is a need, and we can fulfill this need. But again, we have to have access to the people understanding why and how. So now we have all the data. So I think we can penetrate better the brain of biotechnology people.
Yeah. So for me, so there was a survey among the like 20 biopharmaceutical companies. They're the major player in the biopharma field. So they're asking the usage of polysorbate in their formulations. Actually, all of them concerned about the polysorbate usage, especially their stability and their fertility because they have to do a sterile filtration for their final products. So the loss of polysorbate during the filtration, and also along the storage, the degradation of polysorbate products, which cause the formation of particles, do concerns them. So there's a need, an actual need in the industry to find a alternative or better solution to tackle this issue with the polysorbates. Yeah, as Carmen said, we have a solution, one of the solutions that can kind of solve this issue in the formulation for the biologics. But we need to let more people know, to understand, and at least take a try first.
Threading on that, along as we are talking about polysorbate stability, we had some questions about cyclodextrin stability. So how do you measure the stability of hydroxypropyl cyclodextrin? What kind of analytical tools you have to do that?
Okay, so thank you for sharing this question. Actually, I also want to add up a little bit that's regarding the last question you mentioned, and Carmen and Peng also addressed a lot of questions regarding that protein formulation. So actually, whatever is new, the chemical apply in a pharmaceutical industry, they always come with a challenge. Is if you want to replace the new component in a formulation, then you need to address the analytical strategy and the challenging, because each of the simple component you change, then you need to fully develop each of the analytical strategy, which is a lot of work, and sometimes even not able to finish this kind of the task. I guess there's some kind of the challenging we are facing now. We are lack of the validate and what is that supposed to say is kind of the very comfort the method are able to use to determine the concentration as well as its individual component of that the cyclodextrin. So because from the pharmaceutical manufacturer point, if you wanted to, that concern some the safety or toxicity, the issue in a formulation, you need to have exactly chemical information. And without this information, then there's a lot of questions cannot be solved very well. So I guess that's the question we need to address. And we recently also received a lot of requests from the pharmaceutical company. Even they are very keen to use that cyclodextrin. So their lack of the analytical strategy are able to, for example, to measure a lower quantitation of the cyclodextrin in a formulation in a different, like a upstream or downstream process. And I guess another question is regarding the formulation. The Carmen mentioned clearly what is the mechanism for the cyclodextrin as a protein stabilizer in a formulation. We actually have no idea, because generally we saw it offer hydrophobic interaction. Other said will be hydrophilic interaction, but based on our study, which is not always the case. So we can say probably different antibody, different protein, they have a different binding set with the cyclodextrin. So which cause probably will create additional question, because this is not very ordinary phenomenon can be found. For example, we say hydrophobic interaction, we can predict it, there will be work some kind of the class of the antibody or the protein. But the question is, cyclodextrin itself, the chemical structure is very unique. So the different antibody, actually the amino acid sequence will be different. Then will be cause some different when we do the evaluation, how the HPBCD can prevent can offer some the benefit in terms of the protein stability. So all in all, this is I think put all these things together regarding that that the chemical, the complexity. So then to go back to the question you have regarding what is the technology so far we're able to use to identify, quantify, and calculate the cyclodextrin. Because this is molecule basically is quite unique, is hydrophilic, a little bit hydrophobic, a little bit. So in terms of the analytical science, you want something either more hydrophobic or more hydrophilic, because we need to develop a unique purification strategy before you do that detection. So most of the time, we can use that most widely the analytical study, which is chromatogram. So chromatogram, either way you choose hydrophobic interaction, carbohydrate, or you use like hydrophilic interaction or the charge band. But unfortunately, cyclodextrin itself not close either of them. So which is that force us to look for the new model of that separation, the mechanism-based separation technology. And beyond of this separation technology, and also there's the difficulty we are facing, which is detection, because this is basically sugar. So the UV detector normally used for a lot of chemical we are measuring, not able to have the signal on the UV detection. Some other technology, very universal one, for example, the RI detector, they have a lower limitation because the detector itself only able to find the reflective the difference, which is need a higher concentration, are able to cause a signal shift. So yeah, so go back to the question is, we face a lot of analytical challenge, not only the chemical itself, the complexity, and also the chemical molecule itself. We face that separation and the detection issue. So so far we did sort out several of the separation technology based on the chromatogram and also the detection technology. We are using the CAD is charged aerosol detector from the Thermo Scientific. So this is the new technology is basically based on the chemical itself are able to form the droplet. So which is doesn't matter the chemical itself has a UV absorbance, has the fluorescence property. So you always can get the signal and also offer the quite sensitive the method we are using. So in terms of other technology, maybe you want to use the LCMS able to detect a low amount of the cyclodextrin, which is an ideal to use that. Why? Because cyclodextrin has more than 21 actually modified site. So basically, you're supposed to see at least 15 or even more substitutes of the cyclodextrin molecule. So in terms of that, the MS detector, you're still not able to get a real information because it's very hard to quantitate each of the component of that molecule. But like I say, this is the mainly challenging for us at the group. You were trying to push cyclodextrin into a formulation area. So we are also looking for the analytical technology help us to quantify the cyclodextrin. Yes.
Thanks a lot, Long. Very comprehensive answer. And just one quick question shortly for Tao on the protein topic, as I just want to have a few minutes left for other topics more in general about biopharma. So Tao, we have a question. What is a typical concentration for protein stabilization of a cyclodextrin?
Okay.
There is a typical concentration.
Yeah, so we suggest to try the range of 5% to 10%, which is equal to like about 40 millimolar to 80 millimolar in the liquid solution. This is considering the efficacy. plus the consideration of osmolality and the viscosity that the molecule can introduce to the solution.
Okay, thanks a lot.
So it's a
Thanks.
5 to 10% will be the optimum concentration range.
I have a question for LM. So today we talked a lot and most about gene delivery and proteins and protein delivery. How about other uses of cyclodextrins in biopharma, in cell therapy, in vaccines? How do you see the future of these, again, new applications in your experience or feedback with your partners?
Yeah, Thomas, I think with Milo's presentation, very nice stuff, I would say, which give a very promising future for cyclodextrin, modified cyclodextrin. So it's not only actually cyclodextrin is a great tool for drug delivery system. Okay, the most important is the question, what do we want to achieve? The first question, what do I want to achieve with my modified cyclodextrin? Because if I achieve the performance that I have set up at the beginning during my development, I have my product quality target profile, I got it. But now what's about the safety? Because this is coming to what Carmen was saying, biopharma, and we know it, are very conservative, you know? They if something is working, they continue on it. Why should I change? Because if I change, I have to re-explain safety and so on. This is clear, and we fully understand this. Like for polysorbate, they know there is an issue with polysorbate. Everybody knows there is a lot of publication, but they continue with it because it's working. And the thing with the modified cyclodextrin, there is a promising future, not only with a gene therapy, with cell therapy, with vaccine, with any, if I'm talking about biological, as long you achieve, if you achieve the performance, I would say the functionality of your modified cyclodextrin, you achieve the performance in vitro, now you need to show it in vivo, and most important, you need to still demonstrate the safety of use, no immunogenic reaction, no interaction with other component of your body. This is really clear for everyone, but all this, there is a lot of challenge, and we are really happy to see that a lot of academic, like Cyclodextrin, a lot of pharma company are still working. There is a lot of challenge, and everybody wants to improve efficiency, yield of production, because these are really costly manufacturing. So yes, there is a promising future for modified cyclodextrin in biological field.
Yeah. And of course, we haven't talked today about such applications, but we all know that there are cyclodextrin, specifically hydroxypropyl beta cyclodextrin in vaccines like the Johnson & Johnson vaccine, also in clinical studies and as vaccine adjuvants, cryoprotectants, also research done in antibody CD conjugates. So there are indeed quite a lot of applications. Last question before we conclude, I guess it's mainly for Milo, but anyone can jump in. So would it be possible to make cyclodextrin-based carriers to improve delivery throughout the blood-brain barrier? Not necessarily focusing on proteins or on genes, but if you can relate your answer to this, that's even better.
I mean, that would be beautiful. It would be, I don't know, the golden egg. A lot of big pharma and everyone is curious to and eager to achieve an effective cross of the broadband barrier. There are different approach. I mean, the one I'm more, let's say, expert about is the targeting. I mean, there are specific receptors on the blood-brain barrier, and there are a lot of publications. We did also some study about that to modify cyclodextrin to cross effectively. I reported some example in the literature where my idea that, okay, they target some receptor, the GLUT receptor, the whatever receptor is expressed on the blood-brain barrier or the SRV, what we were showing during the presentation can be effective. It's complicated. It's really complicated, but in my opinion, it's only a question of time. It's only a question of time and money, of course, but it can be achieved. So we have to be positive always about this one, no doubt.
Okay, our time is up, but I think that was a great session. So I'd like to thank all of you, Carmen, Elam, Caitliona, Tao, Eilong, Milo, for the great opinions and the great talks. We have addressed most of the questions, but the ones that we missed out, I promise to take offline so we can talk about that. Thank you everyone for attending. I hope that you enjoyed the webinar and that we could provide some new insights and useful information. And otherwise, of course, all the speakers, all of us are available to provide further information for your questions. So thank you all for being here today. I enjoyed it a lot. I hope you did too.
Thanks so much. Thanks to you. Thanks everyone.
Thank you, everyone.
Thanks.
Thank you for your time.
Thanks.
Have a good day.
Thank you.
Bye-bye.
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 Bye-bye.